Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Right now you are watching Bioinformatics Insights. If you are new to this YouTube channel, so let me tell you that we have started this series computer-aided drug designing, and today we are going to cover part 8 of this series. So it's a kind of skill-based course. If you have not subscribed yet, so I would suggest to subscribe this channel. You will receive many academic videos in the coming days or in the coming months. So let's start. Today's tutorial is all about CLIFs. What actually is CLIF? CLIF it stands for Kinase Ligand Interaction, Fingerprints and Structure. Why we are covering this tutorial and how much important is this tutorial, we will discuss. So far we have covered all these topics, and we have covered not only the literature point of view, but we have covered that how we will be able to do it practically. So I will not discuss them anymore. And in part 7 we have covered like how to calculate off-target binding proteins for a drug. But today we will cover kinase ligand interaction, fingerprints and structure, what we call CLIFs. Okay, let's start. So here is the CLIFs database, clifs.net. You can open on your computer. What is all about this CLIF? You can see here kind of a phylogenetic tree, and it has 1 2 3 4 5 6 7 groups, and these are the name TKL, TK, etc, etc. So let me tell you that there are 518 kinase proteins working in a human system. If the number is so high, it means that these kinases are almost working everywhere. They're working in cancer, they're working in different types of cancer, they're working in almost all the pathways. Okay, if you are new to biology or if you are not so sure, then I have also covered a video in initial days dissecting protein research, but I will comment one point from there, that if you listen a word or if you heard a word kinase, so you should technically know that the kinase means that this protein will perform a function of interacting ATP and then takes one phosphate and will relieve ADP, and that phosphate normally will work for its activation. We call it that the enzyme is not active. So in overall scenario, if there is a kinase, its primary substrate will be ATP. It will start its function after recognizing the ATP. So there must be a pocket which interacts ATP. Now if we combine these two statements, that there are 518 kinase proteins working in the human system, and they all are different, but the biology is working on homologs. From homolog, I mean that there is an evolutionary relationship. So, if a protein belongs to a kinase family, and particularly if it is in this group TK, so it is expected that the binding pocket will be mostly similar of all those kinase proteins, and the same goes for other groups. So this database, to what we call it CLIFs, it actually collects the information of all the kinase protein whose structures are available in the PDB. Also, this database provides the information of those inhibitors or those drugs which are attaches to those kinase proteins. For example, in this course we are always working with EGFR, so I will check EGFR. So if I go, yes, it's here, epidermal growth factor. So if you can see, it belongs to the TK group, and these are the PDB available, these are the chains, and also EGFR family, it is a kinase, and if there is any ligand attached here, like you can see its information, these are the ligands which are attached with these different, and all are tested in humans. So it's a long list. You can download the structure, download the CSV or Excel, or you can work with CLIFS database. Come here, clifs.net. But we will learn to work from the Google Colab, and we will also learn how to use this database in our drug designing project. So coming to this part, how you will open this part, it's the same thing, just I will drop the link. Let me repeat it again. I will drop this link, you will open here, and once you see this kind of thing, then you need to select CADD project, and in CADD project come explore CLIFs in kinase research part 8 here. Once it is open, so you will be required to connect. Okay, now here is a brief introduction about the channel, about myself, and about the CLIF. Okay, what will be our aims in this tutorial? So we will access this database, or I would say we will access CLIFs database, and we will calculate some fingerprints, we will take some structure, we will use those fingerprints, or this way we will be able to understand our receptor or the active pocket of our receptor further. Okay, these are some references. This is about some kinases, and I have already discussed this. Okay, in today's tutorial I will discuss once again EGFR, but this time I will give gefitinib. Gefitinib is once again a kinase inhibitor, and just like imatinib, you will find many kinases attached to gefitinib. So then next we will here we can find some information about the CLIF database and its role and its documentation and its literature if you want to study. Here it is. Okay, Now here we will start some installation stuff, but once I start it, so I will pause the session not to waste your further time. Okay, welcome back. We are done with our installation stuff. It will take almost 5 to 4 minutes. Now we will define kinase and ligand of interest, EGFR and gefitinib. In your case, you have your own kinase and you should have your own inhibitor. So it is very easy. First just search your kinase here. You will find out the group name. Also, if there is inhibitor, you will find out that thing and all other related information. So species human, kinase group TK, kinase family EGFR, kinase name EGFR, and ligand expo ID is IRE. If you are going with gefitinib, so just type gefitinib in this drug section or in the search box, so you will get its ID, that is IRE, just like STI for imatinib we use in our previous section. Okay, so you will ask the program to remember these stuffs. Now we will generate some API. We call it API, and okay, here we ask the program just like to search the CLIFs database and tell me how many groups are there. So 7 I showed you on the tree a little earlier, but they are 8 because the other is also here, which cannot be grouped to any of these 7, but still they are kinase, so they are grouped in others. Okay, then we will ask the program that how many proteins do you have in the TK family or in the TK group. So we ask in this command like kinase group to give us this information for this kinase group. What is this kinase group? We have already decided here, like the kinase group should be TK. In your case it will be different. Probably in my case it is TK. Okay, then next, how many kinases belong to this group? So it just prints some of them, so it's the information, maybe not very much important for you. Okay, now 406 EGFR structures are present in the TK group. We ask here the program just to search for the kinase name and tell me how many do you have. So the kinase name in the first command, we already asked this notebook, like the kinase name will be EGFR, so we have 406. Now we are asking like how many structures do you have available. So we got information in this kind of thing, and total 549 structure for EGFR, only EGFR are available. This is the pocket information for the EGFR, and this is some resolution, RMSD, RMSD2, and quality score. So these are some relative information you will get. Okay, then we ask the program to display the pocket sequence. So these are actually 85 residues. You can see here that they are the pocket residues for EGFR, and they are interacting in different drugs with different drugs. Okay, now here we are calculating the fingerprints. We call it the fingerprints. We already talked so much in the part 1 or 2, I guess, when we were dealing that Max and Tanimoto fingerprints. Now we generated those fingerprints in the kind of mathematical way. If certain fingerprint is available, so it will be 1. If not available, it will be 0. So the computer can understand it. So you don't need to understand these fingerprints, just like 0 or 1. Then we are able to get the structural coordinates, just like the information of the PDB file. And we can also get the information about the IC50 and the PIC50 from the Kimball database. Okay, so this was all about how we can use CLIF. But now in further section we will use a kind of case study on EGFR. So we will install, so far it is not installed above, the biopandas. So after installation we will be able to perform some analysis using the EGFR protein. Okay, you can clear it. We will use the OpenCAD package. We import some essential libraries from the OpenCAD, and then we are calling the structure from the CLIF database. What we did now, get interaction fingerprints for the EGFR structures. So in your case it will be your structure, in my case it is EGFR. So these are all the interaction fingerprints. I just show you 5 or 6 entries, otherwise it's a longer list. Then we are going to average the interaction networks, which this command has done it. And we will click this one, okay. These are just a quick overview what kind of interaction fingerprints we received from the CLIFs about EGFR. And it's a longer list, but we have print only 5 or 6. Now we will generate some figure. Yes, it is. So these are those 85 residues which are a kind of pocket residue for EGFR, and which interaction they normally make or they normally form with inhibitors. So from the color you can read them, which residue will make which interaction most of the time, just like the blue color is hydrogen bond acceptor. So you can see the 48 residues. It can make 3 types of interaction: the hydrogen bond as a receiver, here is a donor here, and it can also make apolar contact, but normally it will be, I just assume that in form of the apolar contact, it will use its backbone atoms. Okay, now we are asking the program that keep only those structures which has bound ligand. So it has just printed all those structures. Now we have it says that the structure of EGFRs, which contains IRE. IRE, I told you in the start, in our case it is gefitinib. In your case it could be a different protein and different one. So we have at 23 structures available. We are trying to preview this on the NGL view. Okay, here is the NGL view, the gefitinib with the EGFR. It's just for the image, you can render it. I will not run it, that's okay. So now let's highlight residue which shows frequent interaction with ligands or with inhibitors. So I just selected here like hydrogen bond donor or hydrogen bond acceptor. Both are forming, these both features are for hydrogen bond. Runtime disconnected. Run it again, connecting. Okay, they have just the same figure but in a closer view. Okay, show all kinase bound ligand using the RDKit function. So these are the all ligands, not only the IRE or gefitinib, which are bound to EGFR. In your case you will have your own ligand, and there are total 149 ligands, but we have placed or we have print 10 of them. If you want to print more, just change this number to that number you want to. Okay, now here is coming the interesting part. We will use Kimball and will receive the data from Kimball about these structures which are bound to different ligand, and then we will plot it here. And if you can see, like more than, this is the bioactivity standard, so more of them the frequency is higher than 200 for IRE and nanomolar activity. It means the gefitinib is pretty much fine as an inhibitor so far with EGFR. You can read these things because I'm going a little faster, because someone has also messaged me that my videos are sometimes unwantedly longer. Okay, it's almost the same thing, but we have refined our data specifically to the EGFR. Now we are going to explore some random kinases in CLIFs. Now here we will see 10 ligands from those random kinases. But they were not 10, so I can see only 3, but that's okay, it's not important. And the last one is to show molecule mole complex in PDB. Okay, so what we got from this notebook? From this notebook, we actually became able like how to find out the interaction panel, or how to find out the interacting pocket or interacting residues from the CLIF. Basically, this notebook was designed that we should learn how we can incorporate CLIFs using the Google Colab. In the coming video, we will combine the off-target part 7 and this CLIF bioactivity, or the interaction fingerprints, and we will analyze some real data. Okay, so far it was part 8. I hope you have learned that what is CLIF and how it will help you in your drug research. And don't worry, as I told you that 518 inhibitors are there. So if you are busy in some drug research, so 90% plus your receptor will be a kinase. So you will need, or these kind of notebooks will help you. So let's wait for part 9, and there we will just explore how to combine the part 7 and part 8, and we will perform some analysis. That's it for today. Thank you everyone, and once again a request, if you have not subscribed this channel, please subscribe and share it in your relative groups, so that at least I should be motivated. Okay, thank you everyone, bye bye.